T lymphocytes can recognize specific antigens on human tumors. Tumor rejection antigens are peptides of tumor cell proteins that are presented to T cells by MHC molecules. These peptides can become the targets of tumor-specific T cell response because they are not displayed on the surface of normal cells, at least not at levels sufficient to be recognized by T cells. 6 different categories of tumor rejection antigens can be distinguished. The first category consists of antigens that are strictly tumor-specific. These antigens are the result of point mutations or gene rearrangements which often arise as part of the process of oncogenesis. Point mutations may evoke a T cell response either by allowing de novo binding of a peptide to MHC class I molecules or by creating a new epitope for T cells by modification of a peptide that already binds class I molecules. A special class of tumor-specific antigen in the case of B and T cell tumors, which are derived from single clones of lymphocytes, are the idiotypic sequences unique to the antigen receptor expressed by the clone. The second category comprises proteins encoded by genes that are normally expressed only in male germ cells, which do not express MHC molecules and therefore cannot present peptides from these molecules to T lymphocytes. Tumor cells show widespread abnormalities of gene expression, including the activation of these genes and thus the presentation of these proteins to T cells. Hence, these proteins are effectively tumor-specific in their expression as antigens. The third category of tumor rejection antigen is comprised of differentiation antigens encoded by genes that are only expressed in particular types of tissues. The best examples of these are the differentiation antigens expressed in melanocytes and melanoma cells. A number of these antigens are proteins involved in the pathways of production of the black pigment melanin. The fourth category is comprised of antigens that are strongly overexpressed in tumor cells compared with their normal counterparts. An example is the HER2-NEU, which is a receptor tyrosine kinase homologous to the epidermal growth factor receptor. This receptor is overexpressed in many adenocarcinomas, including breast and ovarian cancers, where it is linked with a poor prognosis. MHC class I restricted CD8 positive cytotoxic T lymphocytes have been found infiltrating solid tumors overexpressing HER2-NEU, but are not capable of destroying such tumors in vivo. The fifth category of tumor rejection antigens is comprised of molecules that display abnormal post-translational modifications. An example is the underglycosylated mucin MUC1, which is expressed by a number of tumors, including breast and pancreatic cancers. Proteins encoded by viral oncogenes comprise the sixth category of tumor rejection antigen. These oncoviral proteins are viral proteins that may play a critical role in the oncogenic process and, because they are foreign, they can evoke a T cell response. Examples of such proteins are the human papilloma type 16 virus proteins E6 and E7, which are expressed in cervical carcinoma. And that's T lymphocytes can recognize specific antigens on human tumors. Have a great day.